By way of introduction and disclosure, I'm Graham Ross. I'm a full-time employee of Roche Pharmaceuticals, and I'm the clinical science leader for the development of an anti-HER2 antibody called pertuzumab. And I'll use pertuzumab as an example of this pathway to registration. These are the themes that I'll cover during my talk. I'll describe neoadjuvant therapy in general and pathological complete response in particular. With this background, I'll then speak about the evolution of pathways to registration of new active agents in early breast cancer. The development and registration of new agents is an expensive and time-consuming process. It takes many years. But these are essential steps to making the medication available to patients. And in order to improve outcomes overall and to enable patients to have access to new active agents, any measure that can be used to expedite this process is highly desirable. Just for orientation, in this schema, one can see the different therapeutic modalities commonly used in treating early breast cancer. Typically, the diagnosis is made by breast cancer screening or breast self-examination. The disease is then properly staged to make sure it's definitely early-stage tumor. Surgery is and remains the backbone of therapy. But according to the tumor characteristics, the estimated risk of relapse, and local treatment guidelines, there are various therapies used as adjuvants to assist the surgery in ensuring that the tumor does not relapse and optimizing the chances of good long-term survival. So much for adjuvant therapies. What is neoadjuvant therapy? It's reasonable to give any or all of the adjuvant treatment prior to the surgery. And when this is done, we call it neoadjuvant therapy or primary systemic therapy. Note that in clinical practice, anthracyclines, the anthracycline component of the typical adjuvant chemotherapy regimen, are not given in combination with the anti-HER2 antibodies because of concern for cardiotoxicity. So they're offset, and that's reflected in the diagram. You see a small offset between the antibody treatment and the chemotherapy treatment. It's been shown that if one uses neoadjuvant treatment, there is no compromise of patient outcomes. If the same treatment is given before surgery or after surgery, the outcomes are the same. This meta-analysis, published by Maury from the National Cancer Institute, is one of several that give us this reassurance. But if the outcomes are not improved by neoadjuvant therapy, why would one do it? These are the potential advantages associated with neoadjuvant therapy. None of these points can be considered proven, but all are supported by evidence. And I suspect that only the first will not be controversial. So, neoadjuvant therapy is the treatment of choice for inflammatory breast cancers. But in addition, shrinking the tumor with neoadjuvant therapy is believed by many to facilitate the surgery in the ways listed. Neoadjuvant therapy also enables response-assessed treatment. So if the tumor responds to the therapy, one knows it's efficacious, one continues. If the tumor does not respond, one can change the treatment. And that reduces the exposure of a patient to an inactive therapy. One can also assess biomarkers during the course of the treatment, such as Ki67, and assess the response to the treatment given. And so neoadjuvant therapy is ideal for research, particularly biomarker-driven research. In addition to these advantages, for those patients who experience complete pathological response, as described by Dr. Sahin earlier, there is a consistent and repeatable association with good clinical outcomes. And so it's reasonable to suppose that if we can add new agents to the neoadjuvant regimens and increase the rate of complete pathological response, we will ultimately improve the outcomes for patients. So what does complete pathological response look like? And Dr. Sahin has given us some very good indications. But the fundamental pathology criteria for defining complete pathological response were formalized by a group of pathologists from Aberdeen. They described, based on retrospective data, 5 levels of response to neoadjuvant therapy. A level 1 response is no change, or no response at all to that therapy. And in the photomicrograph on the left, you can see sheets and sheets of malignant cells. There is no response to that treatment. At the other end of the scale, one sees complete pathological response or an absence of invasive tumor from the primary breast tissue. These criteria, or variations of these criteria, are widely used, and they become known as the Miller-Payne criteria. The point of this is that the assessment of pathological complete response is not technology dependent, and I think that's really important in the context of registration. Registration of a technology is a whole new ballgame. But the pathology in any clinical study needs to be carefully thought through before embarking on that study. Why is pathological complete response important? Whoops. These are the outcomes shown retrospectively by Miller-Payne. Because they're retrospective, they cannot be considered proof of a correlation between PCR and outcomes. But the evidence of the association is striking. And you can see that those that have a level 1 response, or no response at all, have a very poor outcome. Those that have a level 5 response, a complete pathological response, have a very good outcome. These data from Romero, published in the Annals of Oncology in 2013, corroborate Miller-Payne. So there are 3 different expressions of PCR shown. Of particular relevance are C and F, and they show overall survival at the top and relapse-free survival at the bottom, a very good independent corroboration of Miller-Payne criteria. And just incidentally and importantly, Kaplan-Meier curves A and D corroborate the residual cancer burden as described by Dr. Sahin earlier. So we're seeing very good corroboration of the different ways of assessing a pathological complete response. Over time, different interpretations or definitions of the Miller-Payne criteria have evolved. Very importantly, each of these definitions has been shown independently to be associated with good long-term outcomes. In the second column of the table, you can see that absence of invasive disease from the primary breast specimen, or breast PCR, is actually the same as Miller-Payne level 5 response. And this definition has been used by a number of research groups, as you see listed in the bottom cell of that column. As Dr. Sahin has said, to have a really good outcome, one should have absence of invasive disease in the breast and the axillary nodes at surgery. That is known as total PCR, and that's described in the middle column. And that, again, has been used by a number of important research groups and is the preferred definition used by the regulatory authorities, including the FDA. The German Breast Group, or GBG, go one step further, and to qualify as a GBG pathological complete response, one needs to have elimination of all the invasive disease and in situ disease from the primary breast specimen, as well as axillary node negativity at surgery. So we see this consistent and repeatable association between PCR and outcomes. But there are limitations. Pathological complete response is not a perfect surrogate. And in fact, formal surrogacy for PCR has not been established. In a particularly important meta-analysis, Pat Cortazar from the FDA and her colleagues attribute this limitation to the heterogeneity of breast cancer. And because breast cancer is so heterogeneous, we have insufficient evidence in each of the specific subtypes to prove that correlation. But I will say, as a personal prediction, I think as we accumulate the evidence in individual subtypes, that correlation, I think, will be established. Importantly, the increase in pathological complete response rate associated with new active agent that is likely to translate into a meaningful improvement in outcomes is not known. But what do we know about the increase in PCR and outcomes? In the first column of this table, one can see a list of therapeutics known to improve survival in breast cancer: docetaxel, anthracyclines, and the anti-HER2 antibody trastuzumab. The increase in PCR rates associated with each of these efficacious agents is shown in the middle column of the table, and you can see that they range from 12.4% to 17.6%. The corresponding disease-free survivals associated with those agents are listed on the right-hand side of the table. And so it does appear that if one has a new active agent which is consistently associated with an increase in pathological complete response rate of about 15%, one would reasonably expect that new active agent to be associated with good long-term outcomes. This is just a reminder that breast cancer is a heterogeneous disease. The gene expression profiles and corresponding phenotypes are illustrated. The original work was done by Chuck Peru and his colleagues, but the principles were described very beautifully in a review by Turner and Jones in the British Medical Journal. But the point that I make is that there is probably no single critical number. A meaningful increase in PCR8 in luminal A, well-differentiated hormone receptor-positive breast cancer, might be very different to a meaningful increase in PCR in, say, HER2-positive breast cancer or triple-negative breast cancer. And moreover, the improvement in PCR that is considered meaningful will also be dependent on the tolerability of that new active agent. And so we need a body of evidence to define both the benefit and risk associated with a new active agent. And turning now to the history of registrations for neoadjuvant therapies. Neoadjuvant therapy itself is not a new concept, and as early as 1924, a surgeon called Jeffrey Keynes was using preoperative radiotherapy to shrink tumors. The use of systemic agents, chemotherapy agents, was reignited in the 1980s. Now, to enable promising new medications to be made available to patients with life-threatening diseases, both the FDA guidelines and the European Medicines Agency guidelines do allow for either accelerated approval or conditional approval, respectively. The guidelines can be found in the websites listed under the title, and I recommend these websites to your reading. They are very, very useful. They're a mine of information about how drugs should be developed for registration purposes, but you can also find all the registration claims about those drugs and the actual licensed indications, as well as the patient information and the assessment reports. They're very, very useful websites. Now, 2 agents have been registered for the use of neoadjuvant therapy within the European Union. And the first is letrozole, the aromatase inhibitor. This was registered in 2012. Very importantly, that registration followed its registration as a treatment for metastatic breast cancer and as adjuvant breast cancer. Trastuzumab, the anti-HER2 antibody, was also registered in 2012. And that neoadjuvant registration also followed the registration for trastuzumab in the treatment of metastatic HER2-positive breast cancer and adjuvant HER2-positive breast cancer. But recognizing the need to expedite the registration of new active agents, the director of the Office of Hematology and Oncology Products at the FDA proposed in an editorial to the New England Journal of Medicine in 2012 that for a new active therapy for which there is a convincing demonstration on complete pathological response, with a confirmatory study running, there is potentially a pathway to accelerated approval. In other words, the neoadjuvant registration could potentially precede the adjuvant registration, thereby making new active agents available to patients several years earlier than has previously been the case. The anti-HER2 antibody pertuzumab was the first therapeutic to go through this new pathway and makes a good case study. Pertuzumab was first registered for use in combination with trastuzumab and docetaxel for the treatment of metastatic HER2-positive breast cancer, and a conventional adjuvant study known as Affinity, or protocol BIG 411, enrollment into that study was completed in 2013. So we have a body of evidence concerning pertuzumab. Within that body of evidence is a study called Neosphere, a randomized phase 2 neoadjuvant study. There were 4 arms to this study, but for simplicity, I've just illustrated the results from 2 arms. And for the 107 patients who received trastuzumab with docetaxel, following 4 cycles of treatment, the breast PCR rate was 29%. And for the 107 patients who received pertuzumab added to trastuzumab and docetaxel, the breast PCR rate was 45.8%. And the difference, as you see, was 16.8%. Now, Neosphere was a pure research protocol and addressed a number of important research questions. But in clinical practice, it's very important that patients get a complete chemotherapy regimen as part of the neoadjuvant treatment to maximize the chances of the patient receiving a PCR. HANA study is a phase 3 study in which trastuzumab, given either intravenously or subcutaneously, was combined with a full chemotherapy regimen. And so HANA study gives us a very good frame of regimen a frame of reference in which to judge the neosphere results. So patients in Hannah's study received intravenous trastuzumab with full chemotherapy. The pathological complete response rates were 40.7%. Now, what happens when we add pertuzumab to trastuzumab and full chemotherapy? This question was answered in a study called Trifina, in which 225 patients were treated with a full chemotherapy regimen with the dual antibody therapy. And as you can see, the pathological complete response rates ranged from 57.3% to 66.2%. And based on this totality of evidence, including this consistent effect on PCR rate, pertuzumab was registered as a neoadjuvant therapy for early-stage HER2-positive breast cancer in the United States in October 2013. The condition of that approval is that the outcomes are confirmed by the adjuvant study affinity. The FDA issued its final guidance on neoadjuvant therapies and pathological complete response in October 2014. And as I've said, these guidelines are a mine of information, and anybody considering a study in neoadjuvant therapy would do well to consider these guidelines, whether the study is intended for registration or not. Similarly, the European Medicines Agency issued draft guidelines in March 2014, and these guidelines follow a slightly different pattern. They're more philosophical than practical, but they do make many of the same points. And I think the best way to deal with disappointment is to prevent disappointment. And by using these guidelines, I think one will go a long way to successful registration programs. Turning now to the confirmatory study. Thank you very much. There are two possible approaches to a confirmatory study, and both sets of guidelines allow for either a single study approach, and as you can see, one recruits enough patients to test both PCR and outcomes in the same study. That study has its attractions, and one would certainly show the correlation between PCR and outcomes, but the disadvantage is it would need to be preceded by interaction studies, and it would be a very large study and take a very long time, but it would be perfectly acceptable. The second way to do it, and it's the way we develop pertuzumab, is to do the interaction and efficacy phase 2 studies as combined neoadjuvant studies, show the effect on PCR, and then separately do a conventional adjuvant study. And this way is both time efficient and patient efficient, and it reduces the amount of risk. So in conclusion, for patients in whom pathological complete response is achieved following neoadjuvant therapy, there is a consistent association with favorable long-term outcomes. And for a therapeutic with a well-established profile, including good tolerability and a substantial effect on PCR, with a confirmatory study running, there is potential for approval with approved conditions in Europe or for accelerated approval in the USA. The confirmatory study could be based on a single study approach or a two-study approach. My personal opinion, the two-study approach is better. But overall, using the new pathway to registration, new active agents can be made available to patients expeditiously. Thank you for your attention.